Hello, everyone. My name is Kyle. I'm a graduate student at the University of Guelph, and today I will present to you some of my thesis work titled Field and Molecular Screening of Bacterial Diseases in Ontario Adapted Beans. This research was conducted at the Agriculture and Agri-Food Canada Research and Development Centre in Harrow, Ontario, under the supervision of Dr. Chris Gillard, Jamie Larson, and Owen Wally. So bacterial diseases are a recurrent problem in bean fields in Ontario. They can lead to crop losses of up to 40% in more severe cases, and they are usually difficult to manage. The most common control measures are crop rotation and the use of copper-based bactericides. Crop rotation is not very effective because bacteria can survive over winter in crop debris, and some pathogens have a very broad host range, being able to infect over 120 plant species. And the copper products, they provide erratic responses, which often lead to copper-resistant pathogens. So this causes a significant increase in production costs in Canada, which has to import certified seed from drier regions in the US, like Idaho, where the drier climate helps to prevent bacterial growth. Therefore, breeding genetic resistance into local cultivars would be the best management strategy, as it would save costs from buying copper products and also from seed imports. So the most frequent bacterial diseases in Ontario are common bacterial blight, or CBB, caused by two Xanthomonas species, and bacterial brown spot, or BBS, caused by Pseudomonas syringae pathovirus syringae. Despite being caused by different pathogens, these diseases have very similar symptoms, as shown here in the picture. And since common blight is better known in the literature, brown spot tends to be misdiagnosed, which made it to be sort of neglected for a long time. So brown spot only gained more attention in Ontario when farmers started noticing disease symptoms in bean fields that were planted with cultivars that were supposed to be resistant to CBB. So this created a demand to better understand the genetic control of these diseases, to see how similar or how different the responses are, in order to breed Ontario adapted cultivars that are resistant to both. So in order to breed these genetic resistant cultivars, we first need to identify sources of resistance in our germplasm. And we've been doing this by evaluating genotypes in the field, and also using some molecular markers that have been shown to be good predictors of disease response. So with that in mind, the objectives of our study was to identify genotypes with high levels of resistance, to correlate the response of brown spot and common blight, to detect differences across market classes and seed types, and to validate molecular markers to be used in market-assisted selection. So for the field trials, we used a randomized block design with 85 genotypes, including 8 market classes shown here on the right, 4 from the Mesoamerican gene pool, which are small-seeded beans, and 4 from the Andean gene pool, which are large-seeded beans like kidney beans. So the design included 4 applications, 2 locations, and 2 years. The fields were artificially inoculated with bacteria using a mechanical sprayer, and the disease response was rated using a 0 to 5 scale, where 0 means no symptoms and 5 means more than 80% of disease leaf area. So this rating scale was converted into the relative area under the disease progress curve. The area under the curve is represented by the blue portion of the chart, and the relative area is basically the blue portion divided by the whole area. And this AUDPC measure was used to compare disease response across different genotypes. So for the molecular screening, we amplified these 3 markers via PCR, These markers were identified in previous studies for aiming to find responses associated with common blight resistance. So we tested them to see if they could be good predictors for disease response in our germplasm for CBP. But also we tested them from brown spot to see if they are associated with a more broad disease response to bacteria or if it's pathogen specific. So we use analysis of variance to detect differences across genotypes, market classes, and molecular market groups. And the ratings for brown spot and common blight were correlated to identify the response patterns. So for the results, this is an outlook of the variance components of the RA-UDPC values. So in the plot, the bars represent the total variance, and the color shows the percentage that each variance component occupied. So the effects of genotype, environment, and genotype by environment were statistically significant for both diseases. The effects of environment and genotype were roughly the same for both diseases, while the genotype by environment interaction was lower in common blight than in brown spot. But this might be due to the larger error portion, as shown here. And here we can see the actual distribution of disease response in these box plots, where the area under the curve values are shown in the y-axis throughout different year site combinations in the x-axis. So the lowest the AUDPC values, the more resistant the genotypes were. For brown spot, the average in 2021 London was lower than the other environments, and for common blight, the mean for Harrow in 2021 was higher than the others. So adding a little more detail, this is the disease response by market class. So bars with different letters are statistically different. In general, for both cases, Andean beings from the Andean germplasm here in the left, they were more susceptible than the Mesoamerican classes. So for brown spot, only 2 groups were observed, one including all the large seeded beans, and plus the small red from the Mesoamerican pool. And for common blight, there were 3 groups, one with all the kidney beans and small red from the Mesoamerican pool, and also black beans. Navy beans were alone with the lowest ratings, and pinta beans had a more intermediate response. So the correlation results for brown spot and common blight responses showed a strong positive correlation for disease ratings, which suggests that there are common loci controlling the disease response for both pathogens. So, and when you highlight the bean types over the plot, we can see how disease response was lower for Mesoamerican beans, which are represented here by the blue dots, while the large seeded beans have higher values here in orange. So, and we can also see some interesting lines that didn't have as good as correlation. So these lines are interesting to study the different response patterns to detect genes that control specific resistance to each disease. And here the same correlation plot, but showing the differences between the different molecular marker groups. So for the marker BC 420, only a few genotypes had the upper band, which is associated with lower disease response, and you can see them in the graph here. They had lower ratings, and the differences between groups were statistically significant. So this is a box plot showing the distribution of genotypes from different groups, and we can see how the genotypes with the upper band, shown here by the number 2, are more tolerant with lower ratings. Here's a second marker. This is the most recent marker developed for common blind, and it also had a statistically significant difference between groups, and we can see that the beings that had like the upper band, shown here in orange, had lower ratings, and this can also be seen here in the box plot with the lower ratings on the right. And lastly, the SAP6 marker, which is the most traditional marker used for predicting resistance to common blight, did not have as much as a good response as the other ones, although it was still statistically significant. So in orange are the genotypes that have this band. This is there's only 2 options for this marker, and you can see that it's all scattered around the plot. It doesn't show us a strong pattern as the other ones. And this can also be seen in the box plot. That doesn't seem to differ much between the 2 groups. So as final remarks, the bean genotypes differed in the response to bacterial diseases. Navy beans were more resistant to bacterial diseases than the other marker classes, while kidney beans are more susceptible. And the bean responses to brown spot and common blight were positively correlated. And the markers, the 3 markers, can be used to successfully to predict disease response to both brown spot and common blight, but more specifically, those 2 first markers. For next step, we're going to focus on breeding resistance into large city beans. So we're going to develop mapping populations with Andean beans to detect QTL, quantitative trait loci associated with disease response. More specifically for brown spot that doesn't have too much information on the literature. Also going to try some genome-wide studies. And the goal is to identify resistant genes and use them to develop and validate new DNA markers to be used in marker-assisted breeding. So I would like to thank the University of Guelph, Agriculture and Agri-Food Canada, and Ontario Bean Grows for funding the research. My advisory committee includes Professor Chris Gillard, Dr. Jamie Larson, and Dr. Owen Walling, and also Dr. Peter Pauls. Thanks. I'd like to thank everyone for their supports, and also all the research technicians and the staff at AAFC that were also involved in the project.